Next speaker is, we're very grateful to have, he must be a very busy man, I'm sure, with all
it's organized for sure.
All of the focus on wastewater testing, now the PCR testing has dropped. And you know, it's really around measuring wastewater, RNA and wastewater as an early indicator. I'll just give you a bit of Dr. Alec Mckenzie's background. He's an attending paediatrician at the Children's Hospital of Eastern Ontario in Ottawa, Canada, has served as the CEO and Science Director of CHEO Research Institute, as well as Vice President of Research for both CHEO and Canada's Federal Genomic Research Funding Agency, Genome Canada. Prior to his involvement in the Ottawa SARS-CoV-2 wastewater surveillance program, Dr. Mckenzie's laboratory has conducted translational research on the rare pediatric disorder spinal muscular atrophy over the past 25 years. In recent years, it has broadened its focus through the Enhanced Care for Rare project to search for therapies for a large number of rare genetic diseases. And I'm sure a lot of people on this webinar will be really keen to hear what you have to say about wastewater monitoring. There's a lot more focus now in that area. So thanks again, and I'll hand the reins over to you, Alec.
Thanks a lot, Kevin. I appreciate that. And that was a great talk. I have to say, of course, I'm a bit more on the optimistic, not as worried as you are as far as the future goes. So maybe we can chat a bit in the panel discussion as far as different world views. But it was a very informative talk for sure. So yeah, I'm a pediatrician and work at CHEO, the Children's Hospital in Ottawa. And about 2 years plus now, my research program ground to a halt, like all of my colleagues, because of COVID. And then my son Duncan called me from Halifax and said he'd seen a talk where it was he saw that SARS-CoV-2 was intestinally shed. We should look in the wastewater. And so I said, oh, that's a good idea. Perhaps we should look in the wastewater. And I went around talking about it for a while. But then Rob Delatello, who really is the leader of this, his wife, who's an art historian major from Queen's University, was nagging him to look in the wastewater. Rob is actually a U of O engineering prof. And so he paid attention to his wife and said he'd start. And we sort of linked up at that stage, Rob and me. But I guess the moral is that listen to your family, listen to your children, listen to your wife. And we started this program over 2 years ago now. I'll try to stay within the 20 minutes here. It's got a fairly rich background, a wastewater-based epidemiology. It blends in with both public health and environmental engineering. Maybe John Snow back in the mid-1800s is the most famous, who he famously removed the pump handle in the middle of London in order to stop the spread of cholera. And that really is pointed to as one of the major public health epidemiologic sort of interventions in a modern time. And, you know, we've seen a lot of progress over the last century, and one would like to think it's us doctors, et cetera, but it has as much to do with sanitation and environmental engineering. And we can see in this graph here the drop of typhoid with water treatment in Philadelphia. And just parenthetically and tangentially, my great-grandfather, Donald Cameron, invented the septic tank in the late 1800s in England. So my return to the sewer may have been sort of preordained in some fashion. The Israelis were the first to use wastewater-based epidemiology for polio in particular. They've had it going since the 80s. They identified a resurgence in 2013. And Canada has their own wastewater program, but it's actually for cannabis and amphetamines that they've been using it for. And it seems that the East Coast is they're more stoners, and the West Coast is more speed heads. It looks like, just based on that data. I'm not sure how old it is. At any rate, that's a program that was initiated by StatsCan about 4 years ago, following the European model. SARS-CoV-2 itself is shed in the intestine. We're not quite sure how it gets there. It goes to the lungs, and it's not in the blood, but somehow it gets in the intestine. We think it might be the epithelial cells might be swallowed and perhaps entrapped in something called exosomes and make it down into the intestine, where it does infect the intestine. And although the actual symptoms are lower, 20% or so, by and large, if you look at viral RNA by PCR, it gets up over 50% or so, over all the way up to 100% as far as a percent of cases which actually will shed RNA into the stool. And just a bit of molecular biology for those that might not know, it's something called reverse transcriptase, PCR. It's different from looking at clinical diagnostics where it's a sort of a yes or no binary result that you get. In wastewater, we need to have a good sense of quantification if we're going to be following the trends in a given community. The potential advantages of wastewater: early warning system, detection of everybody, and the studies have now shown that it really is over 80% of people with the infection who will secrete into the shed into their intestine and then on into the sewage systems. And so as tests come and go, availability of tests, people's anxiety, etc., we all contribute to the wastewater, and therefore it picks up asymptomatic and symptomatic cases. And so we can track it at the level of the population, which is a huge advantage. And the things that we can look at are self-evidently whether it's going up, whether it's gone up and it's coming down, it's peaked and gone down. Kevin, as I wave my arrow around, is that seen by everybody or just by me?
Yeah, we can see it.
Okay, all right. I'll try to point to relevant areas then, at any rate. And so, as one can see, this is the variability one sees in the community testing sometimes, where it's hopefully, in a perfect world, more consistent in wastewater. The first demonstration was Gert van Geens in Holland in 2020. They did this where the cumulative hospital cases were here and the gene copies were here. And one can see, there's a correlation. One can see right away, it's noisy, it's not perfect. So we're asking to do a tricky thing, get a fragile RNA molecule out of a complex wastewater matrix, which it may have rained or the snow may have melted or whatever. So it's a pretty noisy system. It's been used in France in the early days, where a correlation between both the wastewater and the actual cases could be seen. And then there was a case, I think this was in Italy, where basically one can see the viral load in wastewater anticipated the actual reported cases, the red being the viral load here, and the cases seen in the hospital and there. So showing it comes out a bit earlier than the actual case counts do. And so we started in Ottawa about 2 years ago now. And I learned a lot about sewage in the last 2 years, more than I ever thought I would. Given my personal hygiene, my family says it's perhaps not surprising I ended up in the sewer, but I have to say, they're wonderful people, very low egos, the jokes never end. It's been a real revelation working with these people. At any rate, the sewer chain comes from homes, commercial residences, factories, and just rainfall, etc. And it all flows through and ends up in Ottawa in one site, the so-called Rtpcr, or the Greens Creek plant right there. And almost 1 million people contribute to this every day. So in a perfect, it's a great setup. We can go down there and do one test, and that will reflect the COVID level within this entire population. There are shades and nuances. If you live in Munster, it'll take a day and a half to get there. Whereas if you're living in Orleans or Gloucester, it can be there within hours. So while it integrates the entire population, some get there earlier. And indeed, if the RNA breaks down, you may get a stronger representation from this area than you would from Carp or Munster or more distant. But to a first approximation, it's a pretty impressive, high return on investment system, I'd have to say. And so this is getting into the actual shades of of what it goes through at the Greens Creek. The important stuff is up here. This is, I mean, it gets pretty settled and microbial action down there. So we're looking up in this area here. And by and large, there's a filter that gets out coarse bits, and then it pumps through. And then it settles down here, and you've got liquid and you've got solid. And there's a fatty envelope around the SARS-CoV-2, to some extent a nuclear protein envelope as well, and it settles into the solids. And so after much studying here and there, we and people around the world discovered this really was the place we wanted to go in this settled solids area. And once we get it from the clarified sludge sample, there's a bit of concentration. We extract the RNA, and we do this fancy thing called Rtpcr, and then we get the actual signal. Rtpcr, very quickly, you've got the RNA that is floating around the wastewater. There's 30,000 base pairs in every virion. You take an enzyme and you make a DNA copy from that. And so this DNA is going to be the important part. It's PCR amplified. And once you've got the DNA replicated, you heat it, split it, copy it again, et cetera, et cetera. And every cycle, you hear a CT count. Every cycle, you get a doubling. And so we're looking at CTs, cycles of 25 to 35, 2 to the 25th or 35th sort of amplification after you heat, bring it back together, and then elongate at these 3 different temperatures. And these are the PCR primers that we use. This is Patrick, who's praying, Patrick Daou, for success as we started this out. We really were, I think, among the first in North America to be doing this, and we were sort of making it up as we went along to some extent. And in the event, Patrick, who says he's not particularly religious, but in this case, he thought he'd make an exception, it worked out. And a key thing is just chatting with Monir Taha and the other people at Ottawa Public Health, as well as the beautiful people at the actual city of Ottawa who ran the wastewater plant. And they, unlike a lot of public health agencies, agreed to listen to us and talk with us. And at the end of the day, we got a web page up reporting our daily levels, first in Canada, and we think the first in North America, one of the first in the world, where we could actually do a day-to-day sort of revealing of what the signal was. And the number of nurses who came up to me in the hospital saying that their husbands were all over, this is very sort of male-centric, I don't know, but just like a stock market that everybody wanted to watch at some level. And so here you see the reported cases in the green and the actual RNA levels in the red. And it did work out despite all the noise. In the case, there was, back 2 summers ago now, there was a case in the middle here. One sees a new cases spiking in the middle of July. And we got an early warning system around July 15th, July 16th, letting us know that this was coming. And so a 96-hour early warning system there, which we wrote up. If you look at it, there's a number of uses. Obviously, early detection of outbreaks is a key one. Population-wide surveillance, but not only population-wide and detection of early surges, you can go into specific populations. We've done it in prisons, we've done it in long-term care facilities, university residences, northern villages. One is limited by one's imagination. And we also can look for emergence of new variants. As the previous speaker was saying, these are coming along, and we have a way of looking. And I should have had this slide, but we showed in 1 week, from December 12th to December 19th, we just flipped Omicron, the entire wastewater system. Delta went away and Omicron came in. So it's a very powerful window into what's happening on a community-wide level. And we are admittedly seeing BA2 coming in as well, more infective. Not sure it's more severe. The jury's still out on that one. I don't think we can say it's more severe at this stage, but certainly it's more infective. If you have Omicron, you are protected from it from a severity point of view. At any rate, I'm sure we'll chat during the panel discussion. And so these are the various things, and some examples from across the country. Up in the Northwest Territories, they use it in Yellowknife to detect a surge there, an outbreak there. There was a residence in Windsor where they used it to identify and alert the students. And modern buildings all have a place where you can go down and sample it, and there's automated samplers that can take one sample every hour for 24 hours, and you mix them up. That's what we do in Ottawa as well. So you get a 24-hour sort of span. Okay, and then there was an outbreak in a long-term care facility in Hamilton that we were involved in identifying. So basically, it's noisy, and this is the villain right here. And here's the 30,000 nucleotides of RNA. You get a signal RNA molecule per virion. You get literally hundreds of these so-called spike proteins, and as well as membrane proteins, nucleocapsid proteins. So there's more of the protein that is made for every virus. And it's a sturdier undertaking. So basically, wastewater is harsh at the time when the temperature can break down the RNA. Doesn't break down the protein as much. And obviously, there's more protein than virion, 120 and 20-fold in this case. And so we got to thinking, can we go after the actual protein in addition to the RNA? And so we did something called Western blotting, where we actually took proteins from stool as well as sewage, and saw if we could run it down these gels and probe them with antibodies. These are Nafisa and Eamon, who are my graduate students. They're smiling now, but they weren't smiling so much when I explained the project of we're going to look in some stool. But they were heroic, and they did it. And in point of fact, long story short, we identified the protein using standard Western blots here. Then we used a new technique in order to do something called Pcr amplification of the proteins. There's the protein. You get the antibodies. You get two antibodies specific for it. It's got an oligonucleotide DNA tag, and then you have an oligonucleotide that binds these together. And if these two antibodies have bound, you're in a situation where you can actually you have a piece of DNA, and you can do PCR amplification exactly as you do with the RNA, only you're starting with a far greater signal. And so we use it in a small machine called a Fluidigm microfluidic cartridge, where you put little microfluidic spots in there. And then in essence, Nafisa did it, and we compared it. These are all the RNAs, and in the green are the proteins. And long story short, you can see the so-called number of cycles that are needed. Basically, for the RNA, you need 37 cycles. For the protein, you just needed 30 cycles, or 31 cycles. So far more sensitive than RNA. And here's an example of how it works. Here are some cases over from the previous spring. Here's what the basically RNA and protein look like tracking this, whereas the actual RNA itself. So sorry. These are the cases. This is our protein. This is the RNA, which doesn't track it nearly as well. So we really think this is a powerful way of looking at it. We think that it's got a great, 1 to 3,000, 100 to 10 to 1,000 times more sensitive. We can go after 3 targets. Sorry, I got into animation here. I'm not sure why. We can use, a key thing is knowing how much of the wastewater is actual stool. And so we can use 6 fecal content controls, much less expensive than it is RNA. You can do more at the same time, smaller sample sizes. And we're the only one in the world doing this right now, which makes us a little anxious, but we're Tyson Graber at our shop just got some money to chase it down, And so we're quite excited about this approach. So that's it. The next steps, we're broadening this out to looking at influenza, norovirus, something called respiratory syncytial virus. As I said, looking for actual proteins. And one of the proteins that we're going to be looking at as well is antibodies. So we can not only look at the viral proteins, but we secrete IgA, antibodies to COVID and other viruses within the stool. And so we think using this MPAT approach, we can actually start detecting the level of community immunity by detecting the level of spike protein antibodies. Although if you think about it, spike protein antibodies will tell you about how many people have infections and how many people have been vaccinated, because that's what's used for the vaccine. But if you use another protein antibody, that'll tell you just how many people have been infected, because vaccines you don't give nucleocapsid with vaccines. So we think that's potentially a very powerful approach we can use for this wastewater epidemiology. And I'm terrible at pronouncing name, I think it's Grossi, as the previous speaker said, we may not be out of the woods. Right now, there are 7 people in hospital, 0 people in ICU, but this is just the latest fuzzy picture from our website. And one can see it might be going up slightly. It might be. We've come down the peak, and it seems the last couple of days might be going up. Having said that, I really think on a per capita basis, if you really study the numbers, the number of people infected, the number of people who go to hospital, and the number of people who end up in ICU or even dying, it really approximates the numbers of influenza right now, Omicron. At the risk of sounding like a trucker, perhaps, I apologize, but I feel that Omicron is a bit of a different beast, and despite the global mortality, I get worried about talking about danger and talking about keeping us safe. I think that, anyways, I feel that we are comparatively safe. I think our T-cell immunity is very strong. So the chances of getting other infections, yes. The chance of becoming severely ill, I think, is comparatively low. But we can duke it out during the panel discussion. That's just a brief sidebar. This is just showing it takes a village, eh? So this is the Children's Hospital where I work, and all the great people that were there. University of Ottawa, Rob Delatello, whose name, ironically, is not there, is one really running the show. Active Signal in Boston is the MPAT people. The people at Public Health, Munir Taha, thoughtful guy. Rob and I would go on the media and say, it's going this way. And Munir would say, no, you can't say that. And he was very good, very thoughtful, very sobering. And these are the people down at the actual city of Ottawa, as well as the other people across the country that Rob and others have worked with. Anyway, so there you go. Yeah, and happy to do questions now, or Kevin, if you want to just punt along to the next talk, that would be good as well.
Thank you very much, Dr. Mckenzie, and what a fabulous presentation, what fabulous information. So this series is called COVID Science Solutions and Success Stories. And, you know, this is a success story. You must feel so proud of this work. And, you know, just it's phenomenal. So I can probably refer the questions to the panel.
Sure.
But I just thought I'd take license because I'm the host. And I thought I'd just ask you this. So, you know, if we ever get out of this pandemic, if we ever do.
I think panic's a good word. Yep.
Okay. Do you think this testing will continue?
Oh yeah, no, absolutely. Okay. As far as being a success story, absolutely. Rob was invited to the United Nations to present about 2 months ago at a panel. So no, it really has been one of the most successful endeavors I'm involved in. And for me, I got the idea from my son, and then I got a grant to get the money going, and that intellectually was about it for me. But I'm happy to join in and take as much credit as I can. Absolutely. No, the province has stepped up, and I'm remiss not to say this. The province looked at this program. We presented it at the Ontario Science Table, our findings in Ottawa. Peter Uni, the chair, that nice Swiss guy that you hear on CBC all the time, with the sing-song voice, he sort of took it up, and we went to MECP, the engineering department of the Ontario government, and they just ran with it. And now there are 18 centers across the province. We are doing this continually, and it is part of the landscape now. The ground is shifted under our feet, and this is the way things will be done. There's one in Alberta. And so, yeah, Kevin, it really has been like one of the good things, if one can say that, about the pandemic. It's a seismic change in epidemiologic surveillance. But yeah.
Thanks again, Dr. Mckenzie. And really appreciate you sharing this information and your time with us. And if you can be part of the panel at the end, panel discussion.
I'll talk to anybody. Okay, I'll put myself on mute.